Dear students, now in this class we have to discuss about the various cellular components of the immune system. Normally, the cellular components of the immune system are found in the blood. So the immune system is composed of many independent cells which are giving protection to the body against the various microbial infection and also prevent the growth of tumor cells. That's the main function of the immune system and the cellular components. I mentioned already we have the cellular components present in the blood. You know that one we already studied about the composition of the blood in the last year class is being formed of plasma and then the cellular components are the formed cell elements. The formed cell elements include the red blood corpuscles, then we have the white blood corpuscles, and also we have the platelets, the third type. These are all the cellular components present in the blood. RBCs, you know that one, nearly 94% of the cellular components formed by RBCs. 94% of the total cellular components. If we are taking totally 55% of blood is formed of plasma and then 45% is formed of these cells. Out of this 45%, 94% of the cells is being formed of, you know that one, RBCs, erythrocytes. See here we have 40 to 65 lakh cells are present. When compared to the WBCs, the ratio is that one. For every WBC there are about 700 RBCs. If we are taking the ratio 1:700, this is RBC and this is WBC. So the huge number of RBCs are formed in the cells. They are not taking part in the immune functions, immune response. Now we have to go for the second type, the WBCs, the lymphocytes. These are all the cells taking part mainly in the immune response, either in humoral immunity or cell-mediated immunity. That is through antibody production or just directly attacking the cells. Now the leukocytes are classified into two major groups based on the presence and absence of granules in the cytoplasm. One, granulocytes with the granules in the cytoplasm, another one without granules, agranulocytes. Now the granulocytes are further classified into the neutrophils, polymorphonuclear, that is what is called the cells, then basophils and eosinophils. Now the neutrophils are actually more in number, nearly 50 to 70% of the WBCs is formed of, that is neutrophils. Then basophils less than 1% and we have the eosinophils 2 to 5%. Now the agranulocytes, you know that one, we have the lymphocytes and the monocytes. This is what is studied last year. Now among the various types of cells, the lymphocytes are capable of actually producing immune response and also just actually reacting with the antigen. So only we have to study the lymphocytes, the major type, then we have to go for the functions of other types. So the lymphocytes are one of the cells, the only one cells capable of recognizing the specific antigen and they produce what is called the antibodies. Then what is the role of other cells? The other WBCs are involved in three major functions. Okay, the lymphocytes are involved in specific immune response, we can see. The lymphocytes are involved in specific immune response. Now, other WBCs, what is the role of other WBCs? Now, they are involved in non-specific immune response. Non-specific immune response. They are involved in non-specific immune response. Then, antigen presentation. Antigen presentation. I'll tell you what is the meaning for that one. And also the third one, cytokine formation. Cytokine formation. So, lymphocytes are the major cells involved in immune system. Other cells are also involved in immune system, that means other WBCs. They are involved in non-specific immune response. Specific immune response is brought about by the antibodies. Okay, non-specific immune response mostly by the cells, or we have, you know, that the innate immunity production. Now, antigen presentation. Some WBCs are acting as what is called APC. APC. The meaning for that one, antigen presenting cells. I will discuss later what is the meaning of antigen presenting cells. And also the last one, cytokine formation. Cytokine, what is called the chemical signaling molecules released in small concentration. Also I will tell you more about later. So we have non-specific immune response. APC, that is antigen presentation. They are acting as antigen presenting cells. And finally the cytokine formation. These are the three functions performed by what we have other types of WBCs. Now we have to go for the lymphocytes. And before that, you see that all these cells are produced by the stem cells of bone marrow. In the bone marrow we have stem cells. These stem cells are pluripotent. And these cells are called hemopoietic cells. Hemopoietic cells. I mentioned already what you mean by hemopoiesis. The formation and the production of the blood cells is called actually hemopoiesis. So these cells are responsible for the production of the blood cells, hence called hemopoietic cells. They are produced by the stem cells. The cells are pluripotent. So during the early stage of development, the cell is said to be totipotent. That is capable of giving rise to the whole organism. At a later stage of development, each stem cell is capable of giving rise to a particular type of cells and organs or tissues. That is called pluripotency. Ability to give rise to different organs and organ systems. That is called pluripotency. Differentiated into, for example, ectodermal, endodermal, and then mesodermal tissues and organs. Such a type of cell is called pluripotent. So the cells are being pluripotent only at the later stage of development in the case of animals. To begin with, they are totipotent. Say an example of each zygote is capable of giving rise to the whole organism. But later we have formed many cells because of cleavage, resultant cells. They give rise to different organs and organ systems, and that phenomenon is called pluripotent or pluripotency. The cells are called pluripotent and the phenomenon is called pluripotency. So all these cells are produced from what is called pluripotent stem cells, pluripotent what is called hemopoietic stem cells. Each stem cell is capable of producing either what we have the RBCs, or we have what is called the WBCs, or we have the third type, what we have the platelets. Platelets are not considered as the formed elements, you know that one, non-cellular structures. But actually we have certain stem cells giving rise to what we have, that is the platelets. Now let's see what are the different types of stem cells, from which, what are the different types of cells derived. I'll give the flow chart and then I'll go for, that is what we have, the importance of lymphocytes. How the cells are working. I already mentioned about what we have the stem cells. There are two types of stem cells, one embryonic stem cells, another one adult stem cells. And both these cells have the property of undergoing division repeatedly. That's why now the stem cell technology now being used in what is called applications of biotechnology for the production of any organ or even the organism. Now, the hemopoietic pluripotent stem cells are found in the bone marrow. They are called pluripotent. They are giving rise to different types of what is called the stem cells and then finally the cells. Now we can classify the hemopoietic pluripotent stem cells into two categories. One, myeloid progenitor. Another one, lymphoid progenitor. The progenitor means the forerunner, the ancestral cell. That's a meaning for progenitor. Or we can also say myeloid, that is stem cell. Now from the myeloid stem cells, we have the formation of the following cell types. Number one, from the myeloid progenitor, we have cells having larger nucleus. Such cells with a larger nucleus are called megakaryocytes. Now you know the platelets. The platelets are without nucleus, are simply the fragments. taking part in the clotting of blood. Now these thrombocytes, or we can say the platelets, have been formed from the megakaryocytes, which are the progenitor for the thrombocytes or the platelets. As they have large nucleus, the name is given megakaryocyte. Mega means large. Now this is one type of progenitor derived from myeloid progenitor. Now another type of cell that has been derived from the myeloid progenitor, we have the erythroid progenitor. That is a stem cell, also called erythroblast, which normally give rise to the erythrocytes, the RBCs. So the RBCs are developed from erythroid progenitor, that is developed from the myeloid progenitor. This is the base. From that base only we have erythroid progenitor. Some of the myeloid progenitor cells get converted into erythroid progenitor or erythroblast. They undergo differentiation to form the erythrocytes. Now the third type of what is called progenitor derived from myeloid progenitor is the basophil progenitor, one of the components present in the blood. So it is less than 1%, you know that one the least percentage. Now we have from this basophil progenitor of myeloid progenitor, that means the formation of basophils. So we have the formation of basophils from that one, that is basophil progenitor. This is the third type of myeloid progenitor. Now the fourth one, what we have, the eosinophil progenitor. That is also derived from the myeloid progenitor or the stem cell. One of the stem cells of myeloid progenitor. So eosinophil progenitor now give rise to what is called the eosinophils. One with bilobe nucleus. So these are what we call the granulocytes. The basophils and the eosinophils are granulocytes. Here the nucleus is more or less S-shaped. Here the nucleus is bilobe. Now the fourth type over. Now coming to the fifth type. There is one common progenitor for both monocyte and also the neutrophil and another granulocyte. So the monocyte is an agranulocyte structure and the neutrophil is a granulocyte. But there is one common stem cell, what is called neutrophil monocyte progenitor for both the cells have been derived from one common stem cell, what is called neutrophil monocyte progenitor, and which is nothing but a part of myeloid progenitor. So from this neutrophil monocyte progenitor, we have the formation of neutrophils, nothing but the granulocyte. And another one, what we call the monocyte, the largest WBC having what is called the kidney-shaped nucleus without granules. So later the monocytes get enlarged, matured and enlarged into cells what are called the macrophages. So monocytes, from the monocytes we have formed the development of macrophages at the time of phagocytosis. Okay, that's one. Now some of the cells also derive from what is called the myeloid progenitor. And these cells are called dendritic cells. These cells are called so because they have long, thin cytoplasmic or membrane extension. Long, thin membrane extension. You see that one is called long, thin membrane extension. Just like the dendrite of a nerve cell. Hence the name dendritic cells. Now the dendritic cells normally actually responsible for presenting antigen to what is called the helper T cells. Their function is presenting antigen. So these dendritic cells are acting as APC, antigen presenting cells. Antigen presenting cells, presenting the antigen to the T helper cells. I will tell you the mechanism later. Now there are four types of APCs. What are the types of APCs? I am writing here. Number one, what we have Langerhans cells. One type of what is called, that is dendritic cells, Langerhans cells. Number two, interstitial cells. Interstitial cells. Number three, myeloid cells. And number four, that is lymphoid cells. The meaning for that one, these dendritic cells have been formed both from the myeloid and lymphoid cells. After the formation, they have developed what's a long, thin extension of membrane. That's why I mentioned here there are four types of dendritic cells. one Langerhans, another one interstitial cells, third one myeloid, the fourth one lymphoid. So they are called because of the presence of long thin extension, as in case of nerve cells where you have the dendrites are deviating from the cytome, hence the name dendritic cells. So their function, antigen presentation, hence called antigen presenting cells, APC cells. Okay, that's about the myeloid progenitor. Now we have to see the components of lymphoid progenitor. We already discussed the myeloid progenitor, that is one of the pluripotent cells found in the bone marrow, hematopoietic cell. Now another stem cell, we have the lymphoid progenitor. Now it is responsible for the formation of one type of cells, what is called the lymphocytes. Now the lymphocytes are of two types. Some of them are differentiated as B lymphocytes because they are produced in the bone marrow, production and maturation in the bone marrow, hence called B lymphocytes. I already mentioned about what we have, Bursa cells, bone marrow cells. And the other cell types, lymphocytes, which get matured in the thymus gland, they are called T lymphocytes. In the T lymphocytes, we have different types. Some of the cells are called T helper cells. T helper cells, they are mainly responsible for immune responses. Another one, TC cells, they are called cytotoxic lymphocytes, killer cells. So these are involved in cell-mediated immunity, and these cells, the TPG cells, they are involved in humoral immunity or antibody-mediated immunity. Now, at the time of what is called actually there is activation, the B lymphocytes get converted into plasma cells. I will tell you more about while talking later. So B lymphocytes convert into plasma cells, nothing but they affect our cells concerned with the production of antibodies. Then, as in the case of myeloid progenitor, some of the cells are differentiated as dendritic cells. Here too, some of the lymphoid progenitor cells differentiated as what is called the dendritic cells. I mentioned about what are dendritic cells. Now we have to discuss more and more about the lymphocytes. Of all the cells, the only cells capable of, that is actually recognizing specific antigen and producing immune response are the lymphocytes. Recognizing specific antigen and producing immune response are the lymphocytes. I mentioned about the functions of other cell types. We have, they are involved in non-specific immune response or antigen presentation or cytokine formation. I mentioned about, we'll discuss about cytokine. Okay, now, so we have to discuss about the lymphocytes. Now, in the bloodstream, it forms about nearly 20 to 30%. 20 to 30%. Each lymphocyte, so of the total what we have the WBC is 20 to 30% is formed of lymphocytes. They have a long lifespan of 300 days when compared to other cells, the lifespan. Each cell is more or less circular and small, having a large nucleus surrounded by a thin layer of cytoplasm. This is nucleus and this is cytoplasm. Large nucleus and a thin layer of cytoplasm. As I mentioned earlier, we have two types of lymphocytes, B lymphocytes and T lymphocytes. Now, what are the differences between these two cell types? Now, both are produced in the bone marrow. Both are produced in the bone marrow. But the B lymphocytes, after that formation, they undergo maturation. After the formation, they undergo maturation. So, the production and maturation of B lymphocytes occur in the bone marrow. After they get mature, they enter into the circulating blood. They are just actually circulating around the body. So one difference, just common actually origin, both are produced in the bone marrow, though it is T lymphocyte. But the maturation of the B lymphocytes occur in the bone marrow, whereas the maturation of T lymphocytes occur in the thymus gland. That's the main difference. So, after the formation, they are circulating around the body. Some of the lymphocytes formed in the blood and others get aggregated or accumulated in some organs, like for example the lymph nodes and also spleen. Some of the cells are found circulating in the blood, others get normally accumulated in lymph nodes and spleen. About the T lymphocytes, their production in the bone marrow, but maturation in the thymus gland. Maturation in the thymus gland. So that's why they call thymocytes. So just like what is the B lymphocytes, they also get accumulated, that is in the organs, as in the case of lymphocytes, B lymphocytes, the lymph nodes and spleen. So I'll make the difference. B lymphocytes, production and maturation in the bone marrow. T lymphocytes, production in the bone marrow and maturation in the thymus gland. Now, some remains in the blood, others are get accumulated in the lymph nodes and spleen. Likewise, after maturation in the thymus gland, they also get transported to other organs, the peripheral lymphoid organs like the lymph nodes and spleen, as in the case of B lymphocytes. Now what is happening? What is the role of B lymphocytes and what is the role of T lymphocytes? Now, what is the role of B lymphocytes? How is it working? A few words. So normally I mentioned a lymphocyte has a large nucleus and is surrounded by a small layer of cytoplasm, a thin layer of cytoplasm. On the surface of the cells we have surface receptor, nothing but we can say antibody-like substance, attached antibodies. Once when the surface receptor binds with antigen, now the B lymphocytes are activated. So they undergo multiplication rapidly by mitosis to form two types of cells. One, the effector cells, another one, memory cells. The memory cells are long-lived. They retain the memory of the first encounter. They are responsible for the secondary immune response. When the same antigen enters in the body, they just heighten the immune response. So one type of cells, long-lived memory cells. Now these cells do not produce antibodies, but they retain the memory of the first encounter and responsible for the secondary immune response. Okay, when the same antigen enters in the body. Whereas the effector cells doing the functions. Now they form the effector cells, are otherwise called as the plasma cells. They only produce antibodies. So antibodies are produced by plasma cells. If the question comes either in the entrance examination or in our what is called public examination, so antibodies are produced by plasma cells. In both plasma cells and B lymphocytes given, you have to choose only the plasma cells. That is nothing but the effector cell. So that's about what we have actually B lymphocytes and the memory cells and effector cells. What about the T lymphocytes? I mentioned already we have different types. One, T helper cells. It is the one responsible for stimulating the B lymphocytes. So without T lymphocyte, there is no stimulation of B lymphocytes to form the plasma cells. Now the T helper cell, they secrete some chemicals what are called cytokines. What are cytokines? Cytokines are simple soluble molecules produced mainly by TH cells at the time of infection, or also by some infected cells. What is their role? These cytokines stimulate what we have just actually activate the immune cells, particularly we can say the B lymphocytes, and stimulate the process of phagocytosis and cytolysis. This is the main function, phagocytosis and cytolysis. So activate the immune cells and stimulate, not the B lymphocytes to produce antibodies, but stimulate other immune cells responsible for immunity, so that they can just do the action of, that is phagocytosis, that is swallowing the microorganisms or any of the products of the microorganism, and also cytolysis, nothing but the lysis, the breakdown of what is called the damaged cell or the infected cells. Now, the example for cytokines, simply we can say the messenger molecules. Sometimes we can say cytokines are messenger molecules. One such example, interferons, antiviral proteins. Another example, interleukins that take part in tissue necrosis, or what we have, what is called cytolysis, lysis of what is called the cells at the time of graft rejection. While doing the graft, for example, the heart grafting or any other grafting. So other types of lymphocytes, we have so many, that's not given in the book. T killer cells are also called natural killer cells, some meaning, or TC cells, cytotoxic lymphocytes. C for cytotoxic lymphocytes. They are toxic to the infected cells or virus infected cells or the tumor cells. So they move around the body and destroy the damage and also the infected cells. Not only the damage and infected cells, but also they kill what is called the tumor cells. So that's about what we have the components related to the lymphocytes. Now in addition to the lymphocytes, we have also some other types of cells. You know, I mentioned about the neutrophils and monocytes, other cell types. Neutrophils and monocytes. So these are all called abandoning phagocytes. They are doing the function of phagocytosis, abandoning phagocytes. Now what is happening, the monocytes, when they mature, they get enlarged. At that time they are called the macrophages. Mature and enlarge into macrophages at the time of infection. Now these macrophages have, you know that one, free radicals and bacteriolytic enzymes. They perform phagocytosis. But they perform phagocytosis for any microorganisms or anything else that enter the body. Their function is phagocytosis. Largest cell engulfing the microorganisms or any other foreign molecules, any other foreign pathogens, any other foreign substances. That's the role played by the monocytes. So we have neutrophils and monocytes are also doing the function of phagocytosis, and mainly with the monocytes, actually macrophages, engulfing the bacteria and then destroy them. Any microorganisms, any foreign substances. So these two are called abandoning phagocytes because they're always abandoning here and there, just waiting to capture anything foreign that enters in the body and then destroying it. That's about what we have the various components of the immune system, particularly the cellular components of the immune system. So with that I concluded about the cellular components. We have to go for antigen, antibody interactions, vaccines, etc. So I want to do that for the next class. Till then, goodbye. Thank you.